Okay, everyone. Now I'm gonna go over how you can graphically um, represent all the events that I explained previously. So basically, we have what is known as an action potential graph of a skeletal muscle cell. This one is kind of similar to the one that you see in your textbook. Uh, the one that you have in your textbook is of a nerve cell. So there is another one of a nerve cell where you're going to see these line going below the resting membrane potential. But in a nerve cell, the resting membrane potential will be a -70. And so I'll mention it. So basically, what we have here is we're graphically representing, all these steps. And so from -90, we said that -90 was the resting membrane potential of a skeletal muscle cell, okay? And we need to take that -90 to the level of the threshold, okay? That's the event that is happening here in number 1. So these part of the graph, okay, going from -90 to -55, this is what's happening at the motor end plate. So basically, acetylcholine is going to bind to these receptors on this membrane channel. We call this a ligand-gated channel or chemically gated channel in the motor end plate. So once acetylcholine binds there and sodium comes in, then it's going to spread and it's going to create what is known as a greater potential. A greater potential is something that can be measured. So basically, this greater potential, eventually, if we get enough neurotransmitter to bind and enough channels to open, we can drive that resting membrane potential to the threshold level which is needed to open those voltage-gated sodium channels ahead of these ligand-gated channels, okay? So again, these channels, these voltage-gated sodium channels, they're going to open at -55, and that's when an action potential is going to take off, okay? So this is a greater potential, and here we have an action potential, okay? And so notice that now a lot of sodium is rushing in, right? And now we're going to have rapid depolarization, and that's going to drive that membrane potential to +30. Once it's at its peak, okay, voltage-gated sodium channels will close and other channels will open. These are voltage-gated potassium channels that are going to open. So the potassium channels, they open at +30 millivolts. The voltage-gated sodium channels, they open at -55 millivolts, okay? And they close up here. So once you open, once the voltage-gated potassium channels open, then potassium will leave the cell, the muscle cell, okay? It will leave the muscle cell. So that means that if I open the voltage-gated sodium channels and sodium is just spreading forward, in this case of an action potential, the greater potential, sodium is spreading in both directions. But with an action potential, you're going to have sodium spreading forward. But back there, we're going to have a lot of sodium that is going to cause a membrane potential of +30. So there's going to be potassium channels, voltage-gated potassium channels behind, that are going to open, and that's going to lead potassium, that's going to cause potassium to leave the cell, and the membrane is going to be repolarized, okay? Because remember that at the resting membrane potential, it is polarized, so now we're getting to the repolarized state. So who's responsible for this? Potassium. Notice that these potassium channels are going to close at -90. Okay, so that's why you have that curve right there. So it's at that level. With a neuron, the resting membrane potential of a neuron will be at -70. Okay, and since the potassium channels close at 90, we're going to have a state of hyperpolarization. So in a nerve cell, we're going to have a state of hyperpolarization where this curve is going to go below -70. But here, you don't see that, okay? So this is again a graph of an action. This is an action potential graph of a skeletal muscle cell, which is slightly different than a neuron. Okay, how is it different? You don't get a state of hyperpolarization, and the resting male potential is at -90 instead of -70 in the case of neurons. Okay. And lastly, I want to mention that there's different conditions that can affect the neuromuscular junction. Okay, and at the end of the notes, they do talk about conditions like myasthenia gravis, where this is actually an autoimmune condition where your body attacks or destroys these ligand-gated channels. So therefore, they're going to be reduced, okay? And that's going to lead to muscle weakness. In the case of, there's another condition, which is known as, it's also an autoimmune condition, where these voltage-gated calcium channels are going to be destroyed. And this is a condition known as Lambert-Eaton myasthenic syndrome. There are differences between that syndrome and myasthenia gravis, okay, but that's not the point here. Both of them, they're going to lead to muscle weakness, okay. There's another condition known as polio. Poliomyelitis is a virus that destroys these motor neurons, and therefore the muscle is not going to be stimulated, and we're going to get muscle weakness and even muscle paralysis, okay. That condition is going to lead to muscle atrophy. There's another condition known as tetanus. Tetanus. In tetanus, this is caused by Clostridium tetani, okay, and it produces a neurotoxin. We always get a tetanus shot because if we step on a nail that is contaminated with this bacteria, that can cause inhibition of the neurons that are regulating this excitatory neuron. Okay, therefore, if you don't have the inhibitory portion, participating, then this neuron is always going to excite the muscle, and the muscle is going to stay contracted, and we call that muscle spasm. Okay. There's another condition which is called nerve gas. It's used in biological warfare. Where nerve gas binds to acetylcholinesterase, and therefore you have too much acetylcholine available for binding, and that leads to muscle spasm as well. So there are a number of conditions. There's another condition known as botulism, which is transmitted by another bacterium, which is called Clostridium botulinum, and that leads to another neurotoxin that is going to destroy some proteins that are guiding these vesicles to fuse. So those proteins, those guided proteins, are going to be destroyed, and these vesicles are not going to fuse. You're not going to have release of acetylcholine, therefore you're going to get muscle weakness. So there's different mechanisms on which these conditions, act upon, in this neuromuscular junction. The other condition that is mentioned in your notes is Duchenne muscular dystrophy, but that one is a genetic condition that affects males more than females, and it leads to muscle, destruction of the muscle, basically, or production of fibrous, tissue that is going to replace the muscle. So you get a muscle weakness, especially when you're climbing stairs, going upstairs. But it's diagnosed in childhood, and they often die of, in that case, respiratory, failure as a result of affection of their respiratory muscles. But But those are the conditions. We talked about the neuromuscular junction and all of these channels, which are important. That's the physiology of it. And then we talked about the action potential graph, which looks intimidating, but actually, once you study and you know the details, it's not so intimidating, and it's actually fun. On an exam, they can ask you what happens at 1, or what happens at 2, in case there was a 2 here, or what happens over here. Maybe this is a 3 now, and this is a 4, this is a 5. So they have these numbers, and that on 1 we have what is known as a greater potential that develops as a result of opening of these ligand-gated channels. So the resting membrane potential is taken to the level of the threshold. This is a slow depolarization. As these ions, sodium ions come in, they depolarize the membrane, and then we're going to have, once these channels are open, these voltage-gated sodium channels are -55 at that threshold level. Then we're going to have rapid depolarization. This is represented as rapid depolarization. And then at the very top, we see voltage-gated sodium channels that are going to close, and voltage-gated potassium channels that are going to open. Potassium is going to leave the cell, and you're going to take that membrane back to the resting level, okay? And we call that repolarization, because the membrane is polarized when it's resting, when the muscle is resting and it's not being stimulated. So I hope that this helped for you to understand, this information. Okay, so see you in a future video.